Greetings everyone, my name is Saibal and as you are already aware of, I am the current biology faculty from Nucleon Coaching Institute. And this will be my last video for this series that I have started, which was to help you, that is class 12 students, regarding their boards examination with some of the topics that you may find very difficult to understand and which hold a higher weightage. Okay. From this perspective, this will be the last video. Okay, so let's enjoy it. As you are very aware of by now that I have completed my masters from genetics and plant breeding. Okay, with this background, let's start with this. We have already covered all these chapters and all these topics regarding which I had found or I had how to say it, I had gone through an extensive amount of details and extensive amount of hard work to bring these PPTs or slides so that you too can take some photos along with me telling you what these mean. Okay, so we have completed ozone, I guess, yes, and this was the seminar festival. Okay, now something of the applicative nature. Okay, as you see in the PPT behind me, okay, it says that this is an agarose gel electrophoresis. Okay, so what do you understand by agarose gel electrophoresis? First of all, for you to able to understand what is electrophoresis, okay, or what is agarose gel electrophoresis, first you need to understand what is electrophoresis. Electrophoresis by definition simply means that whenever an electric current is passed through some of the organic matters, for example, okay, if it is a DNA, okay, and DNA we all know that it is a negatively charged molecule. Okay, now this DNA molecule, based on the fragment sizes, based on the amounts on which, okay, the electric current can help in the mobility through a medium, this entirety is called as electrophoresis, the splitting or the fragments that move through a lane. Now what is this lane? Lane is that channel or the passage that you have provided by constructing these, okay, and in that electric field itself, okay, in that electric field what happens is, since an electric field must contain a cathode and a anode, the positive and the negative halves. Okay, so can you tell me what can be the movement of this particle, that is a negatively charged particle? Will it move from positive to negative, or will it move from negative to positive? It will be from negative to positive because it is a negatively charged molecule and the negative electrode will basically push it towards the positive side. Now based on the movement after a certain amount of time has passed, okay, based on this movement or mobility, based on this mobility pattern, we get a very distinctive band pattern. Okay, and this distinctive band pattern helps us to understand what is the size of the particles what is the size of the segments of the DNA that you have taken. Okay, this is mostly used or mostly taken in cases where you have to measure from a predefined ladder. Okay, what is this ladder, you may say? Ladder is a predefined how to take a reference. Okay, this ladder is nothing but a reference, okay, from which you will be matching the sizes, because as per this picture itself, okay, you will not be able to understand, okay, now which fragment is going on which, or which fragment correlates to what size. For that reference itself we take the help of a ladder. Okay, and this is when we take a medium, is called as medium based gel electrophoresis, which will be in our case agarose. Okay, now agarose is nothing but a part of this, how to explain, okay, Agarose is a formation of the medium, okay, that we have specifically curated with, okay. We have specifically made it with, okay. For this, when you go in higher classes, you will come to know that we add tris HCl, TE buffer, okay, tris EDTA buffer, on so many things, okay. Agarose itself, okay, to mix it and make it this medium for which the gel electrophoresis should take place, okay. This is the entirety of gel electrophoresis on basis of agarose, okay. Now, as you can see from the power supply, which this is connected to these electrodes. These are what electrodes, and this, okay, this gel tank is submerged. Okay, it is submerged, and this becomes your plate where you will be placing from micropipettes. Okay, from the micropipettes you will be placing it in the lanes. Okay, the specific segments. Okay, where you will be placing the DNA, for which you will be confirming what is their sizes and what is the mobility, whether or not they show polymorphism based on the markers you have selected. Okay, this is a bit glimpse. When you move into higher classes, you will be expected to do these. Okay.